Today, I will be delving into the fascinating topic of transposable elements alongside this article from Science Daily that highlights the importance of transposable elements in regulating genetic expression and advancing genetic revolution. Firstly, let's define what transposable elements are. So, transposable elements are DNA sequences that are also known as jumping genes due to their ability to alter positions within a genome. As explored in the Gen 2052 content, such movement can occur either through the copy and paste method, where transposable elements are copied and the duplicates are inserted at the new site, or through the cut and paste mechanism, where the transposable elements are excised from the original location and moved to a new site. According to the article, transposable elements were once considered as junk DNA, even though they make up for almost half of the human genome. Initially, I wasn't quite sure on how transposable elements were useful other than what I have come across in Gen 2052 that features on how transposable elements are found to have varying levels of activity during evolution, and they have the ability to create genomic instability and also to copy flanking sequences. But through the study, I discovered that transposable elements can interact with the 3D structure of the genome, which hence form variable loops that affect regulatory activity and gene expression in the cell as they create new CTCF protein sites. What personally amazes me in this study is the ability of transposable elements to hijack existing genomes through the formation of new CTCF sites to form new 3D genomic contexts, while creating intraspecies variation and interspecies divergence. However, since the research mainly focused only on humans and mice as specimens while tracking the physical gain or loss of genetic sequences to predict the evolutionary influences of CTCF binding, it could be a limiting factor as we are not able to determine whether transposable elements affect regulatory evolution in all species. In my opinion, transposable elements can be viewed as a double-edged sword because although they present opportunities in fostering genetic diversity, they also possess risks such as causing developmental defects, cell death, or even cancer, as they can mediate chromosomal rearrangements. Moreover, transposable elements are also linked to several genetic disorders such as atypical teratoid, rhabdoid tumor, and neurofibromatosis type 1. This regarding the limitations of transposable elements, I find it exciting to evaluate new insights and future directions relating to the regulatory role of transposable elements. With the vast potential it holds, from having possible applications on neurodegenerative diseases as mentioned in the article, to contributing in gene therapy by correcting harmful insertions, scientists are definitely able to make use of transposable elements in shaping the future of genomics and evolution. I am also glad that I was able to broaden my knowledge regarding gene expression, variation, and evolution, as I was able to explore the limitations and risks associated with transposable elements. It also deepened my understanding on features of transposable elements, which was only mentioned briefly in the Gen 2052 content. Overall, it also made me appreciate the role of these jumping genes more as the ability to translocate is what creates new forms of life.